Unlike complex eukaryotic organisms that grow primarily by increasing the mass and volume of their individual bodies, microbial growth is defined entirely by the population. It is an explosive geometric increase in total cells. A single bacterium elongates, replicates its internal hardware, and splits into two identical daughter cells. To study this proliferation accurately, microbiologists use a batch culture. This is a closed laboratory system, such as a sealed flask or a test tube, where nutrients are strictly finite and metabolic wastes cannot escape into the surrounding environment. This chart outlines the bacterial growth curve, the core analytical model mapping a microbial population over time. It traces four distinct phases: the flat lag phase, the steep climb of the log phase, the high plateau of the stationary phase, and the downward death phase. This curve is not a generalized statistical trend. It is a rigid mathematical output driven entirely by the mechanics of microscopic division and the absolute physical boundaries of environmental chemistry. Looking at the y-axis, we face the central investigative problem of microbiology: how to accurately count an invisible, exponentially expanding population. The first method is direct measurement, which involves physically counting individual cells or colonies. Researchers can use a Petrov-Hauser chamber, a specialized glass slide with an etched microscopic grid, to manually count cells under a microscope. However, without the application of specific fluorescent viability stains, this method cannot distinguish a living, dividing cell from dead cellular debris. To isolate strictly living organisms, microbiologists rely on the viable plate count. Because standard liquid cultures are too dense to process directly, the sample undergoes a strict serial dilution until spreading it on solid agar yields a statistically reliable range of 30 to 300 visible colonies. Over an incubation period, isolated viable cells multiply into these distinct macroscopic structures. This process establishes the unit of measurement as colony-forming units, or CFU, per milliliter. CFU is a metric built on the baseline assumption that each visible colony originated from a single viable cell or an intact chain of cells. The alternative to direct counting is indirect measurement. This bypasses physical counting entirely, correlating the population size to the physical properties of the liquid culture itself. This requires tracking turbidity, or the cloudiness of the liquid, using a spectrophotometer. A spectrophotometer is an instrument that passes a precise beam of light through a sample tube to measure the amount of light absorbed by suspended particles. As the bacterial density in the liquid broth increases, the physical mass of the cells scatters the light beam, preventing it from passing straight through the glass. The instrument's detector registers this drop in transmitted light as absorbance, or optical density. Optical density is a logarithmic measurement that plots linearly against the total physical mass of the cells. Plate counts yield CFUs, quantifying strictly viable organisms capable of reproduction. Spectrophotometry yields optical density, measuring total physical biomass, which includes dead cells. The choice of methodology strictly dictates what the y-axis truly represents. Returning to the growth curve chart, the population line sits flat. This is the lag phase. Cells aren't dormant. They undergo intense metabolic tooling up, synthesizing enzymes for their new environment. Once equipped, cells divide, and the line angles sharply upward into the exponential phase. The interval required for the population to double is the generation time. Because division is continuous, the population explodes via a strict geometric progression, expressed by the equation n sub n equals n sub 0 times 2 to the power of n. That mathematical slope relies on a highly efficient physical mechanism, the strict sequence of binary fission. The sequence initiates when DNA replication begins at a single origin point on the circular chromosome and proceeds bidirectionally. As the newly replicated chromosomes segregate to opposite poles, a critical structural protein called FTSC pinpoints the exact midcell. FTSC is a bacterial protein homologous to eukaryotic tubulin that polymerizes to form a division ring. The FTSC proteins assemble into a dynamic Z-ring at the cell's equator, actively pinching the plasma membrane inward. This Z-ring recruits the divisosome. The divisosome is a protein complex that orchestrates the synthesis of a new peptidoglycan crosswall, or septum, which eventually partitions the envelope to yield two identical daughter cells. The macroscopic, predictable geometry of the log phase slope is directly dependent on the microscopic mechanical efficiency of the FTSE-driven fission cycle. Exponential division eventually hits an abrupt plateau, the stationary phase. In a closed system, essential nutrients become depleted and toxic metabolic wastes accumulate. The environment can no longer support unchecked expansion, and the rate of new cellular division slows until it perfectly offsets the rate of cellular death. As the environment becomes entirely hostile, the culture crosses into the death phase. Viability drops logarithmically, leaving behind a small fraction of highly resilient, slow-metabolizing persister cells. The precise timing and altitude of that stationary phase ceiling are dictated by external environmental constraints. Oxygen presents the most prominent constraint. While molecular oxygen acts as a powerful terminal electron acceptor for energy production, its reduction yields reactive oxygen species. These unstable, toxic derivatives, like superoxide and hydrogen peroxide, indiscriminately damage cellular macromolecules. To survive an aerobic environment, organisms need specific defenses. They rely absolutely on neutralizing enzymes like catalase and superoxide dismutase to dismantle toxic radicals. Thioglycolate medium creates an oxygen gradient to map an organism's tolerance. Aerobes cluster at the oxygen-rich surface. Anaerobes lack defenses and sink to the anoxic bottom. Facultative anaerobes shift metabolic pathways, growing throughout the tube but preferring the top. Similar rigid boundaries apply to temperature. Microbes are bound by cardinal temperatures, minimum, optimum, and maximum thermal thresholds that dictate the structural stability of their proteins. Osmotic pressure acts as a hard stop. High salt draws water out, causing lethal membrane shrinkage, plasmolysis, unless the organism is an adapted halophile. The exact altitude and duration of the stationary phase ceiling is the physical expression of an organism's enzymatic threshold against these extreme environmental gradients. Microbiologists exploit these known physiological boundaries, oxygen tolerance, pH limits, and osmotic thresholds, to engineer highly specialized culture media. Selective media actively suppresses the growth of unwanted microbes by applying precise chemical stress. Differential media exposes invisible metabolic outputs, like acid fermentation, by triggering visible color changes in the surrounding agar. Looking at the fully assembled bacterial growth curve chart, the entire analytical model is visible in context. The precise geometric slope of the log phase is the direct shadow of FTZ-driven mechanical fission. The hard ceiling of the stationary phase is defined by the organism's metabolic threshold against reactive oxygen species and environmental stress, and the ability to plot these phases relies on the epistemological choice of measurement, counting viable colony-forming units or measuring total optical density. The growth curve transforms the invisible, chaotic reality of microscopic cellular division into a predictable, mathematically rigorous, and fully controllable model.